Hello, everyone. Good afternoon if you're on the East Coast. Good morning if you're on the West Coast, like Jenny is on the West Coast. I have Dr. Jenny Linden with me today. She works at Cornell Weill,
Weill.
and Cornell Weill, and she is on the neuroscience team there, studying specifically MS, multiple sclerosis. So we're going to try and get into a conversation with Jenny about her background and some publications that she put out, some writings that she's done, and generally background. She's not only smart and obviously a doctor, but she plays volleyball and played volleyball in college, happens to be on my beach volleyball team as one of the better players, if not the best. So I'm excited about today. Jenny, how are you?
I'm good. Thanks for inviting me to do this. I'm excited.
Awesome. I'm very excited too. So when did you decide that this is what you wanted to do with your life?
Being a scientist?
Yes.
Well, being a research scientist, I didn't figure that out until college because I didn't really realize what a PhD was. I've always loved science and have always been a nerd since I was a little kid. So I knew I wanted to do something in science, but I always thought that meant being an MD, like a physician or a doctor. And so when I went to college, I actually, was planning on doing pre-med, and then, got introduced to the professors and, discovered this whole other area of research, which I just totally fell in love with.
And why did you fall in love with the research?
Because it's problem solving, and it's fun, and I think it's really cool, especially when you get to study things on a cellular level. So you're able to ask a question, like, why is this happening? And then you can go ahead and try to figure out what's making this happen, or what's causing this disease, or how to fix something. So just, it really fit in with my personality.
Very cool. So sometimes things change from school to real life work.
Mm-hmm.
Is your real life work what you thought it would be your day to day?
So I actually never thought I would be in neuroscience. I started in grad school and college, I was more focused on infectious diseases. And so it's just funny how the opportunities, I guess, that are presented to you. And so when I was interviewing for a postdoc position, the neurology lab that I'm in now, Dr. Vartanian, we have a hypothesis that MS might be caused by an infectious agent. So it just sort of worked out that my background, even though it wasn't a background in neurology, meshed well with what they were looking for. But I did always know that I wanted to do research that would impact patients, have a positive effect. It's called translational research, making sure that the research that you do on the bench has the potential to help either diagnose diseases, prevent diseases, or treat diseases. So that was my main focus as a career. I mean, I love science, so I could get really motivated by anything as long as it meant I was going to have an impact on patients' lives. Does that make sense?
Yeah, very cool. So what's the most challenging part of your work, would you say?
It changes as you progress. So you start at the bench, which is my favorite part, because it's sort of like playing and investigating. But especially in academia, as you progress, trying to become independent and, starting your own lab, acquiring funding is difficult. And so a lot of your time is spent writing grants. And, it's fun to review all the literature and see what's already been done, and come up with a new question to ask and a new way to answer it. So it's just been a transition, but I mean, anybody in science will tell you trying to secure funding is probably the most difficult part of this job.
Makes sense. So the fun part is in the practicing.
Yeah.
And the most challenging part is trying to get other people to understand what you're doing and get the funding so you can continue to do that.
Yeah. And it's in this political climate, it's very competitive right now.
I imagine it would be, yeah.
Because there's only so much money. There's a lot of great science going on, but it only goes so far.
That brings up an interesting question, actually. How do you differentiate yourself as someone studying this complicated disease from others that are probably doing the same thing? How do you stand out? I mean, do you say that it's, oh, we have this equipment, or is it just we have this theory and our theory might be better than the next person's?
It's more about the data, I think, that you have. We're sort of, I think more people are becoming receptive. There's other people studying MS that think it's caused by an infectious disease, viruses, or toxic chemical insults. But we're sort of unique where we think it's being caused by a bacterial toxin. So people are, it's been taking a while to convince people to get on board with that. But we have some really strong clinical data, which really supports it, where it looks like MS patients have increased exposure to this bacterial toxin versus healthy controls. And I think what separates us from the people who are studying the viruses is that we've demonstrated that this toxin, how this toxin can actually cause the disease on a cellular level, while the other viral studies, and I definitely think viral exposure plays a role in it. And I don't want to sound like I'm diminishing their work, because it's excellent. But their data is showing that MS patients have more exposure to this virus. And if they get exposed to this virus, their risk increases, but they haven't really shown how the virus would actually cause the disease, like the tissue destruction in the brain and stuff like that. So that's how our work differs from other people.
Very cool.
It's a complicated disease, and there's, genetics plays a role, environmental factors play a role. So there's a total possibility that it could be triggered by more than just one thing.
So do you work together with these other teams that believe it's viral or from something that's virus related, or do you guys have more of a contentious, and I don't mean contentious, but let's say competitive aspect to what's happening, and so you don't share as much data?
We're definitely a collaborative lab, and we work more with the other MS labs that are more focused on the immune response, because it's definitely immune mediated. Something triggers the disease, and then there's a abnormal immune response that progresses it and makes it worse. So there is collaboration going on, and we go to conferences and share our data, and get feedback, but it's still it's controversial.
Okay, makes sense. So what is your personal day-to-day workflow like? Do you get to work and right away check on some experiment that you're doing, or are you overseeing people doing that, or how does it work?
Yeah, so I'm transitioning from doing more. I'm still doing bench work, but I am transitioning to where I'm training people and having them do the work as well. But I guess for me, my days are either totally experimental days where you get to lab, you do your experiments, you're working on the bench, under the microscope. So it's most of your time is spent doing that. And then like right now I'm in a writing phase where after you get all your data, you get your story, you need to put it, in a paper and get it published and get it out there. So those days are just sitting at a computer writing, basically. But the majority is definitely bench work. And so it depends on, what type of experiment you're doing. There's in vivo work, which is, animal modeling. And then there's in vitro work, which is cell culture. So it's different. It just depends on what you have planned for the day.
And we've had conversations about this before where you've had to go in maybe on a weekend or something to check on a mouse to see what happened. And you had a certain time period, I guess, a time frame where you had to do that.
Yeah, it's not a 9-to-5 job.
Right. Which is cool. And I mean, it's difficult on you, but I want to get that across, and I want to understand it more to the degree in which it is very physical or very mentally demanding. So sometimes maybe you're doing an experiment that you have to log the changes every hour, and sometimes it's every week or every 2 days or something.
Yeah, you can have time points that, you start an experiment and then you need to check it, 2, 4, 8, 16, and 24 hours. So you need to be around to be able to do that. Some experiments, the time points are days to weeks. And then other times when we get like human samples, you might get them at 4 o'clock in the afternoon and you have to process them that day. So you're there, until midnight, because they're very precious and they're hard to come by. So you just got to do what you got to do.
Whatever the time, right?
Yeah.
So are you doing multiple experiments at once, or because things are so focused, you really only want to, focus your direction and your mental capacity at one thing at a time, or you'll have multiple things going, and so you'll be logging, oh, this one's a 2 hour, this one's a 3 hour?
That's a personal preference. I multitask. So I started in industry, and actually in the manufacturing part of industry. So it was great training for time efficiency and making sure you get everything done, and being able to do multiple things at once. And the thing with the experiments is you do have long incubation, so you can have like hours, or an hour, 2 hour, 3 hours where you're not necessarily doing something right then with the experiment, so you can do something else. It just depends on your time management and a personal preference, because some scientists just like to focus on one experiment. It's whatever you decide and whatever you're comfortable with.
Gotcha. So it could change day to day, experiment to experiment.
Yeah.
Great.
I'm data hungry, so I like to do a lot.
Nice. That's awesome. I mean, imagine that's helpful, because then the more you can collect, the faster maybe that influences the next thing that you do.
Yeah.
It might change your path. So
yeah, definitely. That's one of the things that's interesting about experiments is you have this plan and hypothesis in mind, and then you do the experiment, and you'll get your data for your hypothesis, but you might notice something else that was unexpected, and then you're like, well, why did that happen? And you start traveling down that path. So it's neat.
And before we get into your publication and really what MS is, because I'll have you sort of explain and talk about that too, 'cause maybe not everybody knows exactly what it is. Talk to me about how difficult it is to get something published.
Yeah. So everything's peer reviewed. So that means you write a paper and you submit it to a journal, and the journal sends it to people who are in your field to make sure that your conclusions are supportive, that you perform the experiments correctly, that you're not overreaching. So that happens on all types of publications, and it's a really important part of the process. It makes sure that the science is sound. The difficulty in getting published depends on what type of journal you're trying to get into. So there's something called impact factor for journals. So it has to do with, how do I explain this? About how many people see your publication, and like how it's cited, and like how it's, it's the impact that journal and your article is going to have on the scientific community. So journals like Science and Cell and Nature and New England Journal of Medicine, those are fantastic journals, and they're very difficult to get into. It's a very high caliber. You really have to have, a novel observation, and you often have to have access to technology, pretty advanced technology that not everybody is available. So those are difficult to get into. And then, it tiers down. So, if you have, it's hard to get published, I'm not going to lie.
It seems that way,
because part of the, it gets peer reviewed, and then, they either say, no, this is crap, don't publish it. Sort of crap, you need to do all this other stuff to be able to get it published, or, it's minor revisions, just fix a couple of things. So it depends on the journal. It depends on the topic, like what area of science you're in. It depends on the novelty of your data. So like if people haven't observed this before, then that's the other thing. It has to be novel to be published, but there's different, I guess, levels of novelty. And it's difficult too, because you can do a lot of experiments, and we call it negative results, where it's, you did the experiment correctly. You have your positive controls to show that your actual setup is working. And then you have your negative controls. So that's like, yes, this causes something to happen. So we know that our experimental setup is correct. And then a negative control that won't show that result. And then you have, your experimental conditions that you want to test. And sometimes they're just all negative. And it's not that the experiment didn't work. It didn't give you the outcome you were expecting. And that data is still really important. But that's hard to get published, because people want to see, it's more exciting if you have a positive result.
Like this is what's going to happen, and then it happened, as opposed to, surprise, we got something else.
Yeah, exactly. Like, surprise, it didn't happen.
Right.
But I wish there was a better way to get that type of data out, because I feel like a lot of labs repeat the same experiments, and they all get negative results, but it's not published, so there's no way to know that other people have already done it. So it's a waste of, because it's not published, it's a waste of time and money.
Yeah, resources. I'm very surprised you don't have some sort of internal system that shares this information so that people don't waste hours and hours and lots of money and time doing this stuff.
Personal communication between researchers, yes. But, every once in a while, you'll see a publication that's, negative data where, someone performed this experiment and they got a positive result. And then a different lab did the same experiment and got a negative result. And so those get published. But yeah, it's not exciting, I guess, enough to publish that negative data. And I've joked around with colleagues and stuff like that. There should be a journal that's just devoted to that to help save people time and money.
Yeah, or at least maybe an app or some sort of internal server system where people can look things up
Just
and
a
share.
website or something. Yeah.
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 Could be a cool thing for you to lobby for and start, maybe.
Yeah. Yeah. There's a lot of people I think that would definitely support it.
Awesome. So before we get into your publication, let's just talk about what MS is. From my standpoint, it's inflammatory auto response to something going on with the myelin in the body, and I don't know if people even know what myelin is. So I'm just hear your sort of explanation, and maybe as simple as possible. I think this is where we're going to get into some things where we're going to try and simplify as much as possible. I want you to speak your mind and say what you need to, and I'll ask any questions to simplify things for me and whoever might be listening. But what is your definition of MS exactly?
So like you said, it's a demyelinating disease, and the immune system definitely plays a role. So it's like a it primarily affects the brain and spinal cord, so your central nervous system, and it's neuroinflammatory. So the things that it specifically targets, like you said, is the myelin. So you have your nerve cells, right? And they're sort of similar to wires. And the nerves are wrapped up in what's called myelin. And the myelin helps the nerves function properly by sending signals, to other nerves and to other parts of the body. And so in MS, this myelin gets destroyed. So that means the nerves don't function properly. They have a problem, sending their signals. And so this can lead to, cognitive disabilities, most often physical disabilities, because your brain and spinal cord, help control
Movement.
Yeah. And so what causes the myelin to get destroyed is sort of the controversy. It's still not really known. So if you take out a piece of brain that has an MS lesion in it, you see a ton of immune cells in there. And so that led people to believe that it was an autoimmune disease, that it was the body attacking itself. And that the immune system definitely propagates the destruction, right? So that obviously your immune system is good at fighting off infections and bacteria, but sometimes it gets out of control. And instead of destroying the foreign substances, it starts destroying the host, its own tissues. However, if you look at new lesions, because lesions can be days to months to years old, and they evolve over time. And so that immune infiltration with the immune cells happens a little bit later. But if you look at the lesions that are only a couple days to hours old, you don't see any immune cells really in those lesions. So that suggests that something else is triggering the disease. Is it a virus? Is it a toxin? Is it some kind of inherent problem with the cells that produce myelin? And then lesions can also, so you get these lesions and you lose the myelin, and sometimes they can recover. So the cells that produce the myelin can remyelinate,
And the lesion goes away?
It never fully recovers, but it partially recovers. But that repair mechanism, as people age, stops working. So
it just gets worse with age, like everything else, basically.
yeah. And a lot of the MS research is focused on how to repair that damage. That's what a lot of labs focus on.
Now, is that because something you said before, which is that these lesions sort of start to occur, but your body doesn't start to react to them till later? Is that what I'm understanding? Like the lesion happens, but you don't see the autoimmune response or the immune response around those lesions until later, until they've gotten worse, maybe. So they're sneaky. They kind of
Yeah, yeah, and I should say this is controversial. The study that I'm talking about with the new lesions, not everybody agrees with it. I mean, the main focus is the main thing of what MS is you lose that myelin and you lose the function of the nerves, and it has severe consequences. That's the basic description of, I would say, MS.
Okay. I mean, speaking about controversial things, I think is a good thing, right? If you're studying some different pathway, and obviously if you're saying no one really knows, so we're trying to move in a direction. So, whatever you feel comfortable speaking about, of course, but I'm behind speaking about things controversial. You've made it clear this is not exactly how it works.
Yeah.
But I think that it's important to have those conversations because the same thing happens in fitness, where people will do a study and say like, oh, this is the new thing that works for this. But it's really not that simple.
Mm-hmm.
And it's not necessarily true because you did 1 or 2 experiments on college age individuals doing these exercises. So totally fair, and open to saying controversial things up until what you are.
Oh, I'm fine talking about it. I just want to be clear about it.
So, okay, so we got that down. It's disease where the myelin is affected. And what is...
And the immune system is
overactive.
Yeah, exactly.
Okay. So what does healthy myelin look like? What if someone just has really good myelin, what does that exactly entail? What do those cells look like? What does that sheath look like?
So myelin is actually pretty cool. It's produced by cells called oligodendrocytes in the central nervous system. And so let's see. So can I do this?
Yes. Great. We're going to get a picture for those who are able to watch on live YouTube right now.
I'll try to describe it too. So you have your nerve sort of, which is like a tube. Can you see that?
Yep.
And then the myelin. So if you take the tube and you sort of cross section it, so you're looking at it as a circle, the myelin wraps around and around and around and around and around. It's pretty neat. So I'm trying to think of a verbal description. It's like a toilet paper roll. Oh. So yeah. So like the tube is the nerve, and then the toilet paper is the myelin, just wrapped and wrapped and wrapped and wrapped around.
Awesome. Okay.
So that's what a healthy looks like, and it's different when you look at histopathological things, because you're sort of taking a 3D environment and putting it 2D, and so and there's different areas in the brain that have, like, the white matter is heavily myelinated, and then the gray matter is less so. So there's different areas of myelination in the brain. And if you look at a tissue section, they have different stains. One of them is a blue stain. So in a healthy brain, in a white matter section, everything's blue. And then in a demyelinating lesion or an MS lesion, that blue is very light or gone because the myelin is gone. And so the destruction of the myelin, it's normally tight, around the nerve. And when it starts to, the myelin gets cleared by a certain immune system immune cell that's always in your brain, and it's called phagocytosis. So it's like sort of eating pieces of it as it goes, and the wrapping gets looser and looser, and then you sort of start losing chunks of it over time. Does that
make sense?
Okay.
So the nervous system signal is electrical, right?
Yeah. Yeah.
Electrical first and then chemical? How does it work? And so you're talking about the myelin's wrapping the nerve sending an electrical signal, from my understanding.
It's an action potential. And so the way the myelin helps is it sort of wraps in chunks. So sort of like a train cars. So there's chunks and chunks and chunks. And then the reason the myelin's really important is because instead of having to send the signal all the way down the nerve, it sort of jumps from one opening in the train to the other, and it makes everything, it's much faster that way. So that's sort of the basic role of it, if that makes sense.
And are those synapses, or the synapses are further down the line?
The synapses are at the ends of the nerves, and the spaces in between the boxcars are called paranodes.
Awesome.
So it's just areas where there aren't myelin to help sort of jump that signal along very quickly.
That's a beautiful explanation. I like that. I think it's very clear, and people can picture that, and I think that's what's important.
Okay, good.
So your study is about a bacteria that possibly is linked to this. So let's talk about that bacteria and whatever you think you could share with us.
So the bacteria name is Clostridium perfringens, and this bacterial species is complicated. They produce a lot of different toxins, like up to 30 different toxins. However, the toxin that we're really interested in is called the epsilon toxin. And so that's only produced by C. perfringens type B and type D. And so historically, it wasn't believed that this bacteria was found in humans. The type B and the type D weren't found in humans. They are found naturally in agricultural animals, ruminants like sheep especially, and goats. And what's interesting is this bacteria, it colonizes in the gut. So it actually starts in the gut. You get secretion of the toxin, and then it causes intestinal permeability, and it enters the bloodstream. And that's how it gets up to the brain. And I can talk more about how that happens in a little bit. But in sheep, what's interesting is sometimes they have these huge bacterial blooms of this toxin, of these bacteria. They get huge production of the toxin, and they get a brain disease and symptoms that's much more severe than MS, but very similar. And it focuses again on the white matter. It causes like massive demyelination and tissue destruction in the white matter area. And the reason we think it's more severe in these animals might have to do with the receptor of the toxin. That gets a little complicated. But also ruminants have very anaerobic intestines. So like humans have a linear intestinal system, while ruminants, are multi-chambered. They eat all this plant material that has to be fermented by bacteria for them to actually get nutrients. So it's called the bacteria is anaerobic. So that means they can't really be in the presence of oxygen. So in an animal that's basically this giant anaerobic fermentation, you can get a huge production of this toxin, while in a human gut, it's not as favorable to anaerobic growth, just because of the way the different animals have involved. So that's really interesting, because there's also this sheep MS link where people have observed that in areas with a high population of sheep, there's that increased risk for MS. And so we didn't even start this hypothesis that this bacteria could be causing it. Someone from Australia, was like, well, maybe it's a disease in sheep that's causing MS. And so we've just sort of taken that over and proven some more stuff. But we've started a clinical trial where we've been looking for this toxin in human patients. So we've been looking at fecal samples. I know you're very willing to donate. And so far, we've found the bacteria in about 20% of MS patients, and we haven't found them in any of our healthy controls. And then we've also, we're doing blood tests to test for the toxin as well. And again, MS patients, if you combine all the different diagnostic tests we do, about 40% of patients, MS patients, have this toxin, while it's less than 10% for healthy controls. So that's exciting, but it's what's the word I'm looking for? It's
inconclusive, not enough, or
it's a correlation. That's it. It's a correlation. It's not proof of concept. What I find really exciting about this is the toxin is so specific for the type of tissue destruction that it does. So it gets into your bloodstream, but the blood vessels in your brain are unique compared to the blood vessels in the different parts of your body. So the blood vessels in your brain are a lot tighter. They can't, because your brain is such a sensitive organ, you can't have this free diffusion of bloodborne material into the brain, because it just wouldn't work. So it's very specialized, but because it's specialized, the blood vessels express the receptor for the toxin, while the blood vessels in the other part of the body don't. So the receptor specifically binds to the blood vessels in your brain, it damages them, and it allows the toxin to get into your brain. And then that same receptor is expressed on myelin specifically. So then it goes to the myelin, and it can kill the oligodendrocytes, leading to demyelination. And so some of the publications have, have identified that receptor as the myelin and lymphocyte receptor MAL. But we've also shown using in vitro experiments, again, where that's in a dish, that it specifically kills the oligodendrocytes that produce the myelin, but it doesn't affect, it doesn't kill neurons, it doesn't kill the other cells in the brain. And we've also shown in a dish that it causes demyelination. So not only do we have the clinical evidence that this correlation, but we have this evidence that it causes tissue destruction that's very similar to what's seen in MS. And so, one of the projects I'm working on right now is taking mice and treating them with the toxin to see if that replicates an MS-like disease. And so far, it's all preliminary, but it's very promising. You get lesions that look similar to MS lesions, far more than you see in other MS animal models.
Interesting. When you're treating the mice, are you injecting it and going the blood route, or are you making them eat things where the bacteria would be in the food?
So the best way to do it would be to give them the bacteria. Unfortunately, this type of bacteria doesn't colonize the GI system of the mouse. And so we're working on ways of trying to develop a system to do that. But right now we're just injecting the mice with the toxin. And you can, like you said, you can do intravenous injection, or you can also do what's called an intraperitoneal. So it's just into the cavity of your stomach, not into an organ, just sort of that space that's in there. Do you know what I'm saying?
Not into, just into the cavity, right?
Into the cavity, yeah.
Right, so just for people in general, you're not cutting a hole in your stomach and sticking it into your stomach or your colon. You have a cavity that's housing all those organs.
Right.
Putting it in there.
It's instead of injecting it into the intestines, which is a surgery and it causes a lot of, it's a distress to the mice. And an IV injection or an IP injection is a lot easier on them. But again, there's people in the lab trying to come up with, develop a method to get mice colonized by the bacteria. It's just going to take some time.
Right. So rewinding a little bit, when you first started, you said you would be willing to go into it a little bit deeper. Obviously, over the last couple of years, bacteria in the gut has been discussed almost ad nauseum, because it's sort of the new thing where people are realizing, hey, this might matter in health for a lot of different reasons. You were talking about how the way that this bacteria works, and maybe you can elaborate on other bacteria too. We know the brain-gut connection is very important just because of, let's say, the vagus nerve system, and you house 95% of your serotonin in your gut, and so you have all these very important neurons and just neurotransmitters. But how does this work exactly, where you're saying most of the time, and maybe in a healthy human gut, things stay within those tubes and that sac that's your stomach, and we have acid and all kinds of different things that break stuff down. But if someone has a leak somewhere, maybe a lesion or
in the intestines,
is that how you think that basically it spreads from being in there to the rest of the body?
The toxin itself has been shown to cause intestinal permeability. And the ability of the toxin to get into the bloodstream is a very well accepted process. Exactly how that happens, people still don't know. But like you were saying about the importance of the microbiome in health, it's really interesting, really fascinating. There's been a lot of studies showing that the microbiome of MS patients is different from that of healthy controls. And your intestinal flora definitely plays a role in your immune system. So for whatever reason, some bacteria stimulate your immune system to be more pro-inflammatory. So it's more active. It can cause more damage. And then other bacteria are actually anti-inflammatory. So it reduces the reactivity of your immune system. So it's lots of very important, still a lot to learn, but very fascinating.
Very cool. So off the cuff, what are your thoughts on probiotics and things like that? Because they've become more popular, but when we're talking about intestinal flora, we're talking about things that are alive. And how often do you ingest, let's say, something that's in a pill that's alive? Do you think that those things work, or only cultures maybe in yogurt or dairy? Or
I'm not 100% sure, but I think with probiotics, they've shown that you have to continually take them to sustain that population, that bacterial population. I'm a fan of probiotics. I take them, but definitely more has to be done. More research has to be done in that area. Because there is the possibility, if you have, one of the things we've talked about is, oh, this bacteria is in the intestines of MS patients. Well, let's give them like a probiotic, and maybe some of the other bacteria can outcompete this and get rid of it. So yeah, that's a huge area of research. And it's just really getting started.
Very cool. Yeah, that stuff is very interesting. I think you were the first person to tell me about the fecal transplant surgery.
Oh yeah. Yeah, it's called FMT, fecal microbiota transplant. So it's where they sort of clean out the system of a sick person, using antibiotics or whatever, and then they take fecal material from a healthy person and recolonize the sick person's intestinal system. And what's interesting is there's been a poster that presented this data for MS, and it was only 2 patients, so it's not conclusive at all, but they did that, and the MS patients that received the fecal material from a healthy control stopped having relapses. So
stopped completely
for the time frame that, when that poster was presented. But again, that's only 2 patients. It's not conclusive. But I think FMT has been really successful with treating ulcerative colitis and maybe even C. diff infections as well. And there's places that focus totally on that. There's banks, where you can get fecal material. It's really interesting.
Yeah, it sounds extremely interesting. So you just said something that leads me to the question that how is it different that maybe how do you think differently as a scientist when you read in a magazine or a newspaper or hear someone talk about a new study that maybe leads one to believe they should eat this certain food or do this certain exercise, take this certain medication? What do you look for? Like if you hear the study just off the cuff or you read about it, are you immediately skeptical, or are you more, oh wow, this is probably because it's published here, this is probably a good thing? Or
well, so I'm first like, oh, that's interesting, but I'm skeptical. And so like if I read it and I don't mean this to sound derogatory at all, just like a layperson's magazine or newspaper, my immediate reaction is I want to go back to the scientific publication and see how exactly it was done. Like how many patients did they look at? Was it in mice? Was it just in cell culture? Was it in humans? That's usually where my mind goes. And you got to look at the experimental methods and figure out, are these conclusions really sound? And honestly, most of those papers, you read them, but the scientific ones are usually they're like more research is needed, of course. But yeah, it's an exciting place to start. I always think it's interesting and want to, look it up.
Yeah, interesting, but want to look it up, which is the way that I am too now. But, and I hope to get people to do that rather than just read something and say, oh, this must be the answer.
Yeah, you definitely have to be optimistic, but make sure it's solid, sound work.
Right. When do you get your best ideas? When you're reflecting back on your experiments? Sometimes right before I go to sleep, I have awesome ideas, I feel like, or when I'm driving, which is weird times, but that makes sense because they say when you're not dedicating your full mental focus to something, then it allows you to sort of get into flow state, maybe in something else, and you have better ideas. When do you get your best ideas?
Walking the dog.
Yeah.
I'm not even kidding. I don't know if it's the exercise, if it's being out in the woods. I don't know what it is, or just being in a new environment. But I walk the dog the first thing in the morning, and so I'm already planning my day, and just thinking about stuff. And I think that's when I get my best ideas.
When you don't have good results
Mm.
in your experiments, how taxing is that on you mentally and physically? Or at this point, are you just sort of attuned to, well, it didn't go the way I wanted, on to the next thing?
It depends on the scientist, but you definitely have to persevere and be tenacious. It doesn't bother me. I'm like, okay, what did I do wrong? Let's figure this out. Let's try it again. It's part of the process. It's part of the problem solving process. Other people, it just depends on the person. But
seems like it's just a beneficial thing, I think, as a human to be able to, drop the ball a little bit or be wrong and then just pick it up and keep going. And I would think that that's a necessity for scientists, and certainly what you do.
yeah, I mean, some people definitely get down about it. I mean, it's, some of our experiments are months long. And if something goes wrong or, you don't get the result, you don't get any results, it's discouraging, but you just got to keep going. I mean, you just can't stop.
Right.
I mean, you even if you didn't see any results, you still learn something. That didn't work, so try something else.
The pleasure in the process.
Yeah. I mean, no matter what you do, even if you get bad results, you've learned something, right? And you can use that information.
So what does your fitness routine look like? You were a college athlete, and you still are. Well, you still are athletic, and I want to know what you're doing lately besides walking the dog, if anything, and or what you prefer to do, if you like to lift weights when you're in the groove, or if you'd rather do cardio. What does your fitness routine look like?
It's hard. And it's mostly, I mean, the days are long, but the difficulty comes with the commute, because I commute 4 hours a day. But it's walking the dog, and then I like doing weights with the power blocks. So I do that. Walking the dog is almost a daily thing, and then the weights are maybe 2 or 3 times a week. And then on the weekends, sometimes I go to the gym with Dan, and then it's all about the stair climber, because I love how exhausted and sweaty it makes me feel.
And the climbing, yeah.
Yeah. My fitness routine is not what I would like it to be right now.
Well, it's always challenging, but and you do get your stuff in, which is awesome. And do you think that any of your science background, anything at all, any experience that you've had, even in college, graduate school, whatever, play a role in your belief in exercise in general?
Oh, it's so beneficial. I mean, there's so many studies that, I mean, exercise for MS is huge. I think it's huge for anybody. The endorphin release, all of that stuff. I mean, I just know from personal experience that I feel better when I work out. And I definitely think some of the exercising, even if it's just brisk walking with a dog, plays a role in like stimulating ideas. Yeah, I don't think you need a science background to know that exercising is good.
Not necessarily, but I'm just curious if that maybe gives you an extra push sometimes, because you might think of an experiment or something and say, well, this can help, stave off this situation maybe from happening.
It's definitely feeds into the guilt, because I know the literature. It's like you definitely should be out there doing something. It's funny though, because I really fell in love with science when I learned how skeletal muscles worked. The little walking with the actin and stuff like that. So when I learned about that, I would be playing volleyball, and that's like all I could think about.
Basically actin and myosin acting on each other in your muscles. Okay.
So,
So explain that maybe how you picture that, because you had some very good ways to visualize myelin.
I don't remember it that well, but, and I forget the exact terms, but it's like it's definitely walking. It's like a hook, you pull it towards you, and then another hook, and you pull it towards you, and then another hook, and you pull it towards you. It's like if you were mountain climbing and the mountain was moving instead of you.
Yeah, and what Jenny's referring to is basically how your muscles contract and extend, but it just happens so quickly. But that's what it is. So it's pretty amazing. The human body is just amazing in
It
general.
it really is.
Mm,
Yeah,
yeah, I
that's
mean,
why I'm a nerd.
and quickly, you did mention that exercise is good for MS
Mhm.
patients. Anything that you can cite or talk about in terms of, is anaerobic better than aerobic activity? Um,
I'm not familiar, but, the website I sent you, I think it has, it's for the MS center that I'm associated with. I think they have a blurb about how exercise is beneficial, for MS. So I would check that to see what is the best type of exercise for MS patients, because I'm not a physician, and I'm not familiar with the literature. So I
okay.
would assume that not super stressful exercise would probably be better, just
Right,
to
you don't
keep
want
keep
too
the
much of
mood.
an inflammatory response.
Yeah, I think it's more about, movement, keeping muscle tone, that kind of stuff. But I would check the website. Oh, and the National Multiple Sclerosis Society website, tons of great information there. Um,
Awesome.
so I would check there.
I've spoken to Jenny before about the Wim Hof breathing stuff, and someone that I know that's an instructor specifically that's working with MS patients, because he's in an area which I think we talked about this in Seattle, where it's one of like the hot spots. There's a lot of people with MS.
Yeah, the further you get from the equator, the higher the risk for MS is.
So, and why is that? Because I mean, before you said it had something to do with maybe certain animals, but
Yeah, people think it has to do with sun exposure and vitamin D production. So vitamin D is definitely important in the immune system, and I think MS people, when they get tested for vitamin D levels, most of them have deficiencies in it, and I think a lot of physicians put them on supplements to help with that.
with vitamin D.
So the reason why that happens isn't exactly know, but they think it's sun exposure and vitamin D production.
And that's in a lot of people, right? I mean, what all the research I've done on vitamin D, because again, this is something that, like gut bacteria, that's been huge over the last couple of years, because they're relating having good vitamin D levels to less all cause mortality in
Hmm.
general. So I feel like vitamin D is important for everybody, for everything almost. It's
Yeah.
linked to everything. So d- anything you can maybe elaborate on that? Have there been specific studies you can think of with MS patients, with anything that you might be able to add with why someone should maybe take vitamin D, or the
It,
argument?
it definitely influences the immune system. Why a vitamin D deficiency has a bigger impact on MS patients, I'm not sure. It's probably to do with some of the genetic risk factors. So MS isn't a genetic disease. Like there isn't one gene that causes it, but there's definitely a genetic predisposition that can increase your risk for it. And a lot of those genes are immune genes. So maybe somehow it plays a role into that, but I'm not entirely sure.
What about the thought process? Maybe, because again, I'm not sure, just sort of spitballing ideas here, but myelin, if I'm correct, relies on fatty acids or good
Yeah,
healthy
very fatty. Yeah.
And vitamin D is a fat soluble vitamin, and the nervous system in general is just better off with good fatty acids around. So maybe there's some link there, but
maybe, yeah, I'll have to check the literature. I don't know the answer to that.
Cool.
Yeah,
I'd be curious to know. But I mean, just in my field, one of the things that came up in my research is that, again, vitamin D good for everything, but really good for the central nervous system as well. And I equate that to movement, healthy movement, the desire to move, being better at it. So
I'll have to look that up.
Again, loose, but well, I could keep asking you questions and go down the rabbit hole in a lot of different things. I appreciate your time. One more question for you. What is your personal definition of success?
Oh God. I guess happiness. Yeah, I would have to say happiness. Like if you're happy with where you are, what you're doing, you're successful.
Awesome.
Yeah, because success is it's different for so many different people. Like what makes them feel successful. But if I'm happy, I feel like I'm doing it right.
I agree with you 100%. Great answer. So if people want to follow up with you, find out more about your publication, I guess the best spot is LinkedIn for you.
Yeah, all my publications are on have a link on LinkedIn.
Have a link on LinkedIn. Awesome. Yeah. So if anyone wants to follow up, go check out Dr. Jennifer Linden on LinkedIn, and you'll find links to all of her awesome stuff. I really appreciate your time, Jenny. This was great. Like I said, I want to ask more questions, but let's keep things succinct. I think we got a lot out today, and you've certainly helped me think about things differently, even just the myelin and that sort of train car link thing, which is great. And I think that simple things like that really help people to understand what's happening inside their body.
Okay. Well, I'm glad I could help, and thanks for having me. I enjoyed this.
All right. Well, we will follow up, Jenny, and definitely do this another time, but have an awesome time in Washington.
All right. Thanks. Bye.
Bye.